and yeah, you can go ahead and get started. Welcome everyone to another end of term conference presentation. Our speaker today is known by most of you, Julian Eggert, who's a PhD candidate in the bioinformatics and computational biomedicine major. Julian received a Bachelor of Science in Bioinformatics with a minor in Computer Science from the University of Nebraska, Omaha in 2014. During his PhD candidacy, his primary focus has been on applications for studying genomics using DNA and RNA sequencing. His research involves the use of network inference approaches for characterizing transcriptional regulation in cellular differentiation and disease. So he's defending his PhD dissertation today. So he's at the very last step. As usual, everyone, probably most of you know the rub. He will give his presentation. We'll then open it to questions from the public. The public, the rest of us will be dismissed, leaving behind his committee who may ask additional questions, and then he's dismissed and the committee wraps things up and comes up with a determination whether he has completed his dissertation. So go ahead, Julian.
All right. Yeah, thank you, Bill. So as you mentioned, my name is Julian Eggert, and the title of my dissertation and corresponding talk today is Network-Based Alternative Splicing Signatures of Drug Response in AML. So just an overview of the key topics that I'll be discussing. First, I'll give a background on alternative splicing and transcriptional regulation, followed by an overview of a methodology known as de novo network inference. I will then introduce my first specific aim of my dissertation, which is to develop a framework for characterizing co-expression of complex alternative splicing variants in the form of network modules. And I'll demonstrate this framework with results from an initial use case in which we characterize co-splicing variation across human tissue types. And then in the final section of my talk, I will discuss the results of my second specific aim, which is to identify co-splicing signatures of drug response in acute myeloid leukemia. So alternative splicing is a process that occurs during transcription in which a single gene may produce multiple distinct RNA transcripts. So when a gene is first transcribed to RNA, only specific segments of the gene, known as exons, are included in the final RNA product, leaving behind the remaining segments known as introns behind. And so alternative splicing refers to the fact that the specific set of exons included in the final transcript may vary from transcript to transcript as the gene is being expressed. So this may lead to the production of functionally distinct isoforms, which in turn may lead to the production of functionally and biologically distinct proteins. And so the process of alternative splicing serves alongside gene expression as an important regulatory mechanism for numerous cellular functions, including specificity of cell types and programming apoptosis. And so when we discuss alternative standpoint from a quantitative standpoint, as you'll hear me do today, we typically refer to the total expression of each transcript relative to the total expression of the gene. Whereas when we discuss gene expression, we're actually referring to the sum of expression from all the alternatively spliced transcripts that are being expressed. So this leads me to introduce the central hypothesis of my dissertation, which is that transcriptome-wide coordination of alternative splicing influences therapeutic response in acute myeloid leukemia. And so what do I mean in regards to coordination? Well, in order to properly regulate cellular phenotypes, the process of transcription must be highly coordinated and under tight regulatory control across the transcriptome. So for example, if we look here, in a cellular differentiation process known as hematopoiesis, stem cells undergo cellular differentiation down specific lineages to become various types of blood cells. And so this differentiation process is driven by specific groups of genes being either up or down regulated in a highly coordinated fashion, and hence they are undergoing what's known as co-expression. In addition, this cellular differentiation process is further driven by highly coordinated changes in the relative expression of alternative splicing variants, and is thus undergoing what is known as co-splicing. So this is a fairly well-studied process to an extent. The figure on the right is just one from many studies characterizing the number of genes and transcripts that are being up or down regulated through different stages of the lineage. Now, it's also important to note, however, that if transcription is deregulated and coordination is then lost, this can lead to the development of various disease-related phenotypes, such as cancer. It's essential to be able to comprehensively characterize transcriptional regulation on a systems level in both normal and disease-related biological systems. So how can we do this? Well, given that transcription occurs through a complex regulatory network, we can study co-expression through the use of what are known as gene co-expression networks, where we infer a network of genes from expression data in order to characterize co-expression patterns between genes and be able to study how these genes may interact with one another. So in a more traditional analysis of gene expression, we would actually just test every detectable gene individually to see if that any of the genes in our data set have a significant change in expression between two phenotypes. For example, if we were looking for differential expression between two tissue types or differential expression between a control and a disease group. On the other hand, gene co-expression networks, they allow us to study gene expression on a systems level and can allow us to better identify the cellular pathways that groups of co-expressed genes may be involved in. And now we do this, of course, under the assumption that groups of genes do indeed cooperate within related pathways and related cellular functions. So it's important to understand that co-expression network studies are done through what's known as de novo network inference, meaning that the underlying structure of the network is unknown, and we must learn the network using gene expression from the samples of our data set. So using the data, we infer relationships between all possible pairs of genes using a similarity measure, such as a basic sample correlation or a Pearson correlation. We then store the pairwise similarities in a symmetric adjacency matrix, and then we infer a network in which the nodes represent our genes, and the edges of the network are based on values from our inferred adjacency matrix. And so this is an approach that's been utilized on numerous occasions for characterizing gene co-expression in different systems. So what about inferring a network of coordinated alternative splicing or co-splicing? So inferring a network of co-splicing is actually far less straightforward than that of gene co-expression. Some of this has to do with the biological complexity of splicing, but this also has to do with the current state of RNA sequencing, which is what we utilize for characterizing transcription. But nonetheless, there are a few approaches that have been demonstrated for integrating splicing into co-expression networks. However, when I was thinking about the requirements of a co-splicing network inference framework in regards to the specific biological questions that I would want to ask and that I would consider to be as meaningful, there are a few aspects missing from the previously demonstrated approaches. So what are they? Well, first off, we want to be able to characterize co-splicing of what are known as complex splicing variants. And we want to be able to formulate complex splicing in a manner that's suitable for a module-based approach to splicing. This would then allow us to perform a module-based characterization of co-splicing across different biological systems, and would also allow us to identify co-splicing modules that may be associated with one or more phenotypes of interest. So what are complex alternative splicing variants? So first off, the intuitive approach for quantifying alternative splicing would be to simply measure the expression of each transcript that is being expressed. However, with the current state of RNA sequencing technology that we use to sequence and measure expressed RNA from cell samples, the readout of RNA-seq is in the form of millions of short sequencing reads that are much shorter than the exons from which they were derived, and significantly much shorter than the introns that separate the exonic segments prior to splicing. So given that the alternative transcripts will still share a large number of exons after splicing, there's a relatively large of reads, so these black reads right here, that will come from these shared exons, making it impossible to distinguish the true transcript of origin for each read. Now, in simple cases, like we see in this diagram, this is less of an issue, but in many cases of splicing, the splicing patterns are actually extremely complex, as is the case with this MDM2 gene here at the bottom. And this is just the tip of the iceberg in terms of how complex the splicing patterns can undertake. So looking at this MDM2 gene, we see that we have a large cluster of exons that undergo selective usage in a very complex manner, and methods that would then try to estimate the full expression of the transcripts from these splicing patterns would be highly inaccurate. And then this would be especially problem trying to infer a network. So in a network standpoint, it would be highly inaccurate, and the inaccurate transcript estimation would lead to spurious edges and essentially a false network structure during the inference approach. So previous co-splicing network approaches, they've basically tried to mitigate the transcript estimation problem by focusing on the expression of the individual exons of the gene that will end up varying across the gene due to splicing by their expression. So one approach is to just simply create a network using the individual exon. However, this introduces a large amount of redundancy into the network, along with the nodes of the network having linear dependencies between those exons of the same gene. So other frameworks, they've tried to utilize what are known as aggregated approaches, in which they essentially construct a network of gene correlations based on the gene's individual exon levels. These methods are effective for giving us some initial insights in terms of how co-expression may differ from co-splicing, but they have a few drawbacks. For one, these methods don't fully capture the complexity of the individual splice variants, and they're not able to characterize the coordination of the usage of these complex splice variants. And secondly, these aggregated approaches, they also don't allow for a straightforward approach to quantify splicing at a module level, so we can perform a module-based network analysis. And so what is this module-based approach that I keep mentioning? Well, in a module-based network inference approach, after we infer a co-expression network, we then perform a community detection procedure in order to identify subnetworks of highly connected nodes of the network or genes of the network, which we ultimately call modules. So we can then take all of the genes from a given cluster, and we can summarize their expression to the module level in the form of just a single value. This allows us to characterize relationships between modules representing groups of highly co-expressed genes, and also allows us to identify modules that may be highly associated with particular phenotypes of interest. And so why do we do this? Well, from a technical standpoint, this serves as a data reduction technique. So we know that dimensionality reduction is an extremely useful tool in the field of genomics. And by creating a network of modules, we can reduce the size of our network, which could be made up of thousands of genes, down to a network that consists of maybe only one or two dozen modules. However, this is also extremely useful from a biological perspective, given that we know groups of genes undergo coordinated changes in expression, and that they interact within specific pathways to drive specific cellular functions. And characterizing network modules in relation to phenotypes allows us to identify the pathways that are involved and allows us to study their most central components. So this is an approach that's been utilized on numerous occasions for studying gene co-expression. For example, looking at the figure on the right, this is from a module-based network inference approach using samples from different subtypes of breast cancer. And the authors of this paper, they've actually annotated the modules based on their functions from a pathway enrichment analysis. So looking at the bottom, we have the 11 modules that they inferred from a network consisting of thousands of genes. And then taking these modules, the authors then modeled the expression of each module against the different subtypes of breast cancer. And essentially, this heat map is essentially indicating which modules are being up or down regulated in the different subtypes. So for example, if we look at this first module on the left, which the authors annotated as being involved in estrogen signaling, we see that this module is significantly upregulated in the luminal A subtype. It has moderate expression levels in the normal-like and luminal B subtype, and is significantly downregulated in the basal subtype. And so what this might be indicating are specific pathways that could essentially be turned on or off in different cell types or in different conditions. So trying to utilize modules for co-splicing inference motivated my first specific aim, which was to develop a module-based network approach to study the co-expression of complex alternative splicing variants. So in order to develop such an approach, we need a methodology that provides a comprehensive representation of splicing given the current limitations of short-read sequencing technology. So here at the top, we have all the possible exons that may be expressed for a given gene during transcription. And in a typical RNA-seq experiment, we would take all the reads from a given sample, and we would align these reads to a reference genome. Here, I've zoomed in on exons 6, 7, and 8, showing how the alignment of these reads might look after the alignment step is done. So first off, notice the size of the reads relative to the size of the exons, and also notice the size of the exons relative to the size of the introns, keeping in mind that I've actually flipped the scale for visualization purposes. So actually the introns are actually much larger than the exons between them. Also, notice that most of the reads here in purple align directly to the exons, while a small portion of reads are split across the intron regions. This is done by the splice-aware aligner during the alignment step. So these are called junction spanning reads that are derived from the end and beginning of exons. They occur due to the fact that during sequencing, we're actually sequencing RNA after splicing has occurred, and therefore these intron regions have been removed from the final RNA transcript. Also notice that some of the junction reads from exon 6 are being connected to exon 7, while some of the junction spanning reads connect exon 6 to exon 8. So what these reads indicate is the expression of transcripts that are being expressed in which exon 7 is either included or excluded from the different transcripts. So to characterize splicing of a gene, we can utilize these junction spanning reads to create what is known as a splicing graph, indicating where alternative splicing of the gene is occurring during transcription. So here, we've made a directed graph where the exons are the nodes and the junction spanning reads indicate the paths along the graph. So this is from a single RNA-seq sample where the numbers above all the edges represent the number of reads that are supporting a particular path. And so you'll notice the red and green. The red edges indicate splicing from previously annotated transcripts, while the green, they indicate novel splicing that were discovered during the alignment step. You'll also notice that certain introns are colored red and green. These are a specific type of alternative splicing event known as retained introns. The entire intron is retained in the transcript. So we construct our splicing graph using exons and junction spanning reads. And then we can move through the directed graph, and anywhere that we have a divergent path, either ingoing or outgoing, we annotate it in the form of what's known as a local splice variant, or an LSV, indicating where alternative splicing has occurred. We can then quantify alternative splicing using the LSVs from the graph, where for each LSV, we measure the relative usage of each junction using the number of reads that either support the inclusion or exclusion relative to the total expression of all the LSV's junctions. And again, so the reason that we do this is to avoid the ambiguities when trying to estimate the full-length transcripts using short sequencing reads. So how can we incorporate these LSVs into a network? Well, if you look at the graph, as I've highlighted here, you'll see that many of the LSVs occur within close proximity to one another, with many LSVs either sharing complete junctions or sharing one or more exons being represented by the LSV. So using this group of LSVs together within the same network would lead to a lot of redundancy and also create edges within our network that wouldn't necessarily be biologically meaningful, given that they all come from the same genomic segment of the transcribed RNA. So what we can do, and what I've proposed for this approach, is we can group all of the LSVs that have either fully or partially overlapping junctions and exons into what I call splice variant regions, or SVRs. We can then represent the splicing of all the LSVs within a given SVR as a single value using what's known as singular value decomposition, which is a used data reduction technique. So for example, if we look at this cluster and we call this cluster SVR1, we see that this cluster has 5 LSVs that are all overlapping each other. Since each LSV will always have at least 2 or more junctions, we select the junction having the greatest variability across our data set. We then take the splicing values of the 5 LSVs from all of our samples in our data set, we perform singular value decomposition, And we can then represent the splicing value of the SVR using the first eigenvector, or the leading eigenvector, which is equivalent to taking the first principal component in a principal component analysis, or a PCA. So then we can use these SVRs as features during network inference, which will then be the nodes in our co-splicing network. So to evaluate this framework, I performed an initial analysis with the goal being to see how well this approach can be used to characterize co-splicing modules across human tissue types, and see if the results of the analysis make sense biologically based on what we understand in regards to tissue specificity. So to understand the results, it's important to be able to understand the network inference steps prior to the network analysis. So first, we're going to formulate SVRs using RNA-seq data from the Genotype Tissue Expression Project, or GTEx, which is a large consortium of omics data from dozens of human tissue types. And so here I've selected 10 different tissue types, with 6 tissues being from different brain regions and 2 tissues being from 2 different heart tissue types. And then we also have tissues from lung tissue, or samples from lung tissue, and we also have samples from skeletal muscle tissue. And so here we have some tissues that are distinct but are still highly related, along with other tissues that we expect to be more differentiated from other tissue types. So using the SVRs from each group of tissue samples, we'll then infer co-splicing networks for each tissue separately. So this will leave us with 10 distinct networks. However, each network will still contain the same nodes, but the network edges will all be independent. We'll then identify a set of consensus modules consisting of nodes that are highly co-spliced across all the 10 tissue types, and then we'll use these modules for our analysis. So here we've inferred our 10 co-splicing networks. Each heat map on the left represents the adjacency matrix of network edge weights for each tissue. And then we want to identify groups of splice variants that are highly connected across all the tissues. So we compute a consensus matrix using a minimum quantile approach, where the strength of the edges in the consensus matrix are essentially based on how consistently heavy they are across all the 10 networks. We then subtract the edge weights from 1, which is why the colors have been inverted, and we use the node distances to perform clustering and identify modules within our consensus network. We can then keep track of the modules in our network by labeling these modules using arbitrary color assignments. So here's our consensus module set. So you'll see that we've reduced the size of our network from about 4,100 SVRs to just 13 co-splicing modules. We can summarize the splicing value of each module by performing singular value decomposition using all the nodes from one module basically representing the module using the first principal component, which is similar to what we did when we formulated SVRs from all the LSVs. We then cluster the modules for each tissue, forming tissue-specific module networks. And we can also cluster the modules using all the tissues simultaneously in order to create essentially a consensus hierarchy, which we can use as a reference when we characterize the intermodular differences across the tissues. And so looking at the different networks, we can already start to see some of the tissue-specific differences in terms of the hierarchical organization of the co-splicing modules. For the sake of time, I'm only going to summarize a couple of the key results from the tissue analysis. So if we take a closer look at intermodular variation, here I've selected three of the tissue networks to use as an example. So we have 2 brain networks, hippocampus and cortex, and we have 1 heart region, atrial appendage. And so going diagonal, we see the tissue-specific module networks. However, the modules the order of the modules are all in the same order based on the consensus module dendrogram. So the heat maps at the bottom left, they indicate the preservation of all the edges between 2 networks. And this is based on this first equation on the right. And so we can see that the edge weights are much more preserved between the 2 brain regions and are still preserved, but less preserved in certain modules between the 2 brain regions and the heart region. Now the bar charts in the upper right up here, they represent the preservation of the module connectivity between 2 networks. the module, where essentially module connectivity is essentially the sum of all the edges connected to a given module. And the preservation of this connectivity is given here in the second equation here on the right. So if we look across the bar charts and we look at preservation of module connectivity, we see across the networks, we see pretty high preservation of most of the modules. With the exception is when we compare the heart tissue to the two brain tissues, we see that the module and the this greenish yellow module, the preservation of that connectivity starts to decrease. And here's where we start to see some of the differentiation of the intermodular connectivities between the two tissue types. Sorry about that. And then finally, this last equation here at the bottom, this is a density preservation measure, which is essentially a summarization of the total connectivity of all the modules and the preservation of the connectivity of all the modules across the different networks. And so that's represented by this D value up here. And again, we can see that we have high overall network density preservation between the two brain networks, and that preservation of density begins to decrease as we compare the brain networks to the heart region. So keeping this density preservation in mind, now we can see the here are the density preservation values now across all 10 of the tissue networks. I've also performed clustering using these values where the preservation was converted to a distance. And then so looking at the heat map and the clustering results, we can see that the related tissues exhibit higher preservation of intermodular network characteristics. And so, for example, if we look in the upper left corner, we can see that the brain regions all have greater similarity to one another. And as we start to move away, the preservation begins to decrease into the other tissue types and compare the brain regions to the other tissue types. Here we can see that the co-splicing modules are enriched for numerous cellular functions. So these are the results showing the number of enriched gene ontology terms for each module based on the genes from which the splice variants within the module were derived. So modules may share several different Go terms. They may share several different Go terms. However, all of the modules also contain their own amount of significant enrichment for what would be module-specific terms. For example, some of the most significant module-specific terms are in the case of the green module, which is highly enriched for Wnt signaling. We see that the blue module is enriched for apoptosis, and the black module is enriched for immune response. So we can also characterize the splicing level of the modules in relation to phenotypes. So here we'll select our phenotype of interest being different tissue groups. And so this heat map shows the splicing value of each module with samples grouped by tissue group. And we can see that the modules exhibit tissue-specific splicing differences between all the different tissue types. So the heat map at the bottom shows adjusted p-values from a statistical test comparing the splicing level of one tissue against all the remaining tissues. So for example, if we look at the purple module, we see that the purple module, if we look at the brain samples and the muscle samples, we see that the purple module is significantly downregulated in the brain tissue, and then it's significantly upregulated in the muscle tissue. And so we can use this as sort of a way to evaluate how well we can capture phenotypes at the module level. So just to kind of quickly summarize the co-splicing across human tissues analysis. So here I showed that related tissues exhibit similar intermodular network characteristics, and that these co-splicing modules are enriched for known cellular functions, and they also contain tissue-specific splicing levels. And so this all serves as a proof of concept for demonstration of this framework for doing a module-based network inference of complex splicing variation. Okay, so moving on to my second specific aim, which is the key biological question of my dissertation. My second aim is to characterize co-expressed splicing variants in AML and identify network-based splicing signatures of therapeutic response. So acute myeloid leukemia, or AML, is a cancer of myeloid blood cells in which genetic changes occur in hematopoietic stem and progenitor cells, causing abnormal proliferation. So this in turn leads to accumulation of leukemic cells within the blood. Now, these genetic changes, they can occur in the form of either large cytogenetic events or small gene mutations. And as a consequence, these genetic events lead to a loss of transcriptional coordination during hematopoiesis and leads to the development of the cancer leukemia cell phenotype. So the long-time standard of treatment for AML has been chemotherapy, often accompanied by hematopoietic stem cell transplants. AML patients, however, they're likely to relapse, to eventually relapse after initial treatment and develop into a more aggressive form of cancer. Over the last several years, however, the use of targeted therapy approaches have been developed in which drugs are used to target the specific mutations or pathways that are being effective during AML development. So these can be used alongside chemotherapy, or perhaps they can also be used as an alternative to standard chemotherapy for patients who might be deemed unfit for standard chemo. So despite promising results, there's still a lot of variability in terms of how patients respond to these different targeted therapy approaches. And this has to do with what is the major challenge of AML therapy development. And that AML is characterized by a highly heterogeneous genomic landscape in which we see mutational events from over 1,000 genes across different AML patients, with each mutation that we see in AML being present in only a small portion of patients. And so each patient will typically have maybe 1 or 2 dozen of all of these potential mutations. So my overall goal is basically to try and characterize some of the transcriptional mechanisms in terms of splicing that might be promoting the therapeutic resistant phenotypes that we see in leukemia cells. And so with this network analysis, what we want to try and do is try and identify co-splicing modules that may be predictive of drug response in a number of targeted therapies. So I'm using data that's from the BEAT AML cohort, which is a large collaborative AML research program led by OHSU. It consists of a variety of data types from AML patients, including cytogenetic profiles, mutation calls from whole exome sequencing, RNA-seq data, which I've used for quantifying splicing variants, and it also contains drug response data from cell viability assays from over 100 different targeted therapy types. Okay, so this is our co-splicing module network. We started with a network consisting of over 8,200 SVRs, and we performed network clustering to reduce the size of the network down to 13 co-splicing modules. So in a typical, so the modules are going to be what we're going to use to as features to look for correlations with drug response. And in typical module phenotype significant analysis, in a typical analysis, we could simply just calculate the correlation of each module with respect to our trait of interest. In this case, however, we need to account for the presence of the various genetic events that we already know to have significant prognostic implications and that we know are functionally relevant in terms of patients' response to treatment. And so therefore, we need to model the co-occurrences of both mutational events and changes in splicing. And so we can use lasso regression, which is a linear regression technique that uses L1 regularization or shrinkage. And lasso tries to reduce the number of features in the model by shrinking non-predictive feature coefficients to 0, and then only keeping the predictive ones. So lasso is essentially inherently a feature selection technique. So for each drug that we're modeling, we utilize lasso regression through an iterative modeling technique by splitting the samples into random training and test sets. And then for each training set, we generate 1,000 random samples with replacement of the same size using the bootstrapping technique. And so for each bootstrap sample, train lasso regression using tenfold cross-validation, and then we repeat the procedure through 100 different iterations, selecting a new random training and test set each time. Finally, we then record the frequency to which each feature was selected as being predictive of drug response for a particular drug by each lasso model. So what does this kind of look like? So first off, so we ran these lasso models for about 100 different drug types. And so here is a heat map showing how predictive each mutation or splicing module is for just a subset of the drugs that we actually modeled. So there's a lot going on here, so I'm going to walk everyone through it. So the rows of the heat map represent the iterative lasso models for each drug. And then the columns, they represent the features that were used in the lasso regression models. So all these features were used together in the lasso regression models for modeling against drug response of a given drug. Now, there's no significant ordering to the ordering of the columns, but I've separated the splicing modules to be on the left and the mutations to be stored on the right. And so the mutations are binary features. It's essentially just whether a certain gene had a positive or negative call for having a mutation. And then so the values and intensity, the color intensities of each cell, they represent the frequency as to which the lasso model selected that feature as being predictive for a given drug. So it's the frequency of that feature being selected as predictive across our 1,000 lasso models for each drug. So we can actually just interpret these essentially as a correlation values. So they're basically correlation values in regards to response for each drug. It's just that it's done in a more sophisticated manner in which correlations of each feature also take into account the correlations of the other features of the model. You'll also notice some columns have NA. So columns with NA in the mutations group, they just indicate mutations that were not included in each module due to a lack of positive mutation calls in samples having drug response data for a given drug. So with all the samples that we use to build the network, not all those samples necessarily had drug response data for every drug. And so that's why we see some NA values for some of the mutations that were included. So looking through this heat map, if you look at the group of mutations, one of the columns that we first start to see light up is the FLT3 ITD, or the FLT3 internal tandem duplication. And so we already know FLT3 ITD as being one of the most significant genetic events in terms of both patient prognosis and response rates to a variety of targeted therapies. However, the novel findings of this analysis occur here, where we see a small group of co-splicing modules that we can see are being highly correlated with response to a variety of different drug types. And so the select drugs that you're seeing here in this heat map, these are the drug models in which at least one splicing module had a correlation of at least 0.7 or greater for drug response for a given drug. And so looking here on the left, we can see that the TAN module is the most predictive module for around a dozen of these inhibitors. However, we also see that the blue module, the green module, and the salmon module also have reasonably high correlations for a few other drugs. And the correlations of these modules, we find that they're predictive for drug response, and then they almost always occur with known recurrent mutational events that have already been studied and that we know to have functional relevance when it comes to therapeutic response. So for example, we say that the TAN co-splicing module is highly correlated with resistance to dasatinib treatment, and it co-occurs with the CBFB MYH11 fusion gene, as well as mutations in KRAS, PTPN11, and TP53. We also have a few drugs, nilotinib, rapamycin, and GDC0941, that are highly correlated with the TAN module, but as we can see in the mutation section, we see that the mutational events may only have just moderately correlations with response outcomes. So this is some cases where we're finding that the splicing may be more predictive than some of the mutations that we see. So our most significant co-splicing module in terms of drug response came from the TAN module and its ability to predict response to Venetoclax. So here we're gonna select the Venetoclax model for further evaluation. And so here is a more close-up view of the modeling that was done for Venetoclax response using both mutations and splicing modules. And so the figure on the right is a buzzsaw plot of our model coefficient frequencies, where each colored segment indicates the non-zero coefficient frequency of the feature with respect to drug response to Venetoclax. And so starting with the TAN module, the features go clockwise from highest to lowest in terms of their correlation to Venetoclax response. And then links between the segments, they indicate the frequency in which a pair of features were selected as being predicted by the same lasso model. So for example, we can see that changes in splicing of the TAN co-splicing module co-occurs with the presence or absence of the PML-RARA fusion gene, as well as mutations of the TET2 gene and the SF3B1 gene. And so the SF3B1 gene is I should point out, is a splicing factor gene that acts as a transregulator for making splicing decisions during transcription. We also see that the green module also shows at least minor correlations with venetoclax response. But focusing more on the TAN module, functional enrichment of the TAN module indicated that it's enriched for numerous immune system-related pathways, including neutrophil degranulation. So neutrophils, they're the most abundant type of white blood cell, and they participate in immune response. And actually, similar findings were found in another study that was characterizing gene co-expression modules in a similar approach like this alongside mutations, and found gene co-expression modules also predictive of venetoclax response. And functional enrichment of those modules showed that they were also functionally enriched for immune-related pathways as well. I should also point out that although the TAN module was not specifically enriched for apoptosis regulation from doing the pathway enrichment analysis, looking a little more carefully at the nodes within the TAN module, I was looking at the module hubs of the module, which are the most centrally connected nodes within the TAN module. And many of the splice variants from these module hubs belong to genes that are known to be involved in apoptosis. For example, the SHISA5 gene and the VERT2 gene are two genes that are known to be operational within the apoptosis pathway and are known to interact with the TP53 tumor suppressor gene. And so why do I mention apoptosis? Well, so venetoclax is a BH3 mimetic. It targets the anti-apoptotic BCL2 protein. And as we know, evading apoptosis is one of the key hallmarks of cancer cells. And venetoclax, it tries to inhibit BCL2 activity in order to promote cellular death. And so patients treated with venetoclax, they usually show good initial response to treatment, but typically they will eventually develop resistance to venetoclax over time. However, we do see increased sensitivity with venetoclax when it's observed in combination with other targeting agents. And so here, as a visual reference, I've taken a subset of the most centrally located nodes within the TAN module, and also some of the most centrally located nodes within the green module, which was also moderately correlated to the venetoclax response. And so using the splicing nodes, I've clustered the top most sensitive and resistant patient samples. And looking at this heat map, we can start to see some of the distinct splicing signatures that are distinguishing venetoclax resistant and venetoclax sensitive patients. And so based on what we've seen in previous studies on gene expression, and again, what we're seeing here with splicing, is that the leukemia cells are developing resistance to venetoclax therapy by essentially deregulating apoptosis pathways. And so what we're seeing here within these splicing modules might be that deregulation of coordinated splicing decisions in relation to apoptosis are further driving therapeutic resistance to treatment with venetoclax. So I've demonstrated some interesting results using this co-splicing network framework. Methods like these, however, they're not without potential limitations, of course. So probably the most specific limitation probably comes from the fact that our ability to infer co-splicing networks is dependent on our ability to accurately quantify splicing, given the current state of the quantification methods. So with the graph-based method for capturing complex, these require the use of junction spanning reads, which really only make up about 15 to 25% of our total read library for a given sample. This can depend on the depth of sequencing and the length of those sequencing reads. From each sample, it requires us to set a minimum read threshold per junction in order to confidently estimate the junction usage. And if a sample does not meet the threshold for a given LSV, it's essentially considered unquantifiable. And so what I'm doing where I'm trying to formulate SVVs, it requires that all the LSVs are quantifiable from all the samples in the data set. And so in the analyses that I demonstrated today, we actually took a fairly conservative approach in which we only used LSVs for SVR formulation if the LSV was quantifiable in all of the samples from the analysis. And so what this means is that if just one sample in our analysis didn't have sufficient coverage for one of the LSVs, that LSV is essentially lost in the analysis. And what this might mean in terms of functionality is that if we lose some of these LSVs and then we don't formulate SVRs for these, it essentially just means that that feature wouldn't be included in the network inference analysis. So this could potentially lead to maybe a potential loss of sensitivity if there may be significant splice variants that are at least significant towards your phenotype, but they just might not be included in the network analysis. So there are imputation methods. Imputation methods would try to model some of the missing values. These have been used in co-expression network studies based on microarray gene expression data. These, of course, could have their own issues if the imputations are somewhat inaccurate. And so really where I think the biggest future directions are in terms of this framework is just our ability to improve the modeling of the splicing quantification. And so with the current approaches, we're able to accurately capture some of these variants, but we need a good amount of sequencing depth and preferably longer reads. And I think over time, as we improve the modeling of these splicing quantification, that will improve our ability to infer co-splicing networks. And also, I think if we improve our ability to quantify splicing for unsupervised learning. And so the methods like the MAGIC framework that I use to quantify splicing from these splicing graphs, they're more specifically designed for the two-group analysis of splicing, or similar to just doing differential expression for the genes, where they essentially modeled changes at the group level between two groups of interest to see if a splice variant has significantly changed in usage. And so I think if we develop the methods to be a little more supportive for looking at the splicing values of the individual samples and being able to use them more in an unsupervised analysis, given that de novo network inference is essentially an unsupervised analysis approach, that will improve some of our inference approaches. It'll also improve our ability to work with large heterogeneous data sets where we may have variability in coverage and we may have samples that may be extreme outliers within our sample cohort. So to summarize, I've demonstrated a network inference framework for co-splicing that can include complex splicing variants in a comprehensive and non-redundant fashion. With this framework, we can summarize splicing in the form of modules and characterize co-splicing modules across various biological systems and phenotypes. And I provided results from a proof of principle analysis in which we characterized co-splicing variation across human tissue types. And I showed that related tissues exhibit similar network splicing characteristics compared to non-related tissues. And finally, I demonstrated the results of my second specific aim, finding co-splicing modules predictive of drug response for multiple targeted therapies. And we find that the splicing modules co-occur with mutations that are known to have prognostic significance in AML. And the results of this aim may be revealing intrinsic mechanisms to which the cancer cells may be obtaining drug-resistant phenotypes. And so before I close out, I would first like to thank my dissertation advisory committee, Christina, Guanming, Bob, Ted, and Shannon. I especially want to thank Christina Zhang, who has been my research advisor over the last 5 years and has worked extensively close with me on working on this project. I also want to especially thank Shannon McWeeny, for it was the day that I met Shannon, which was the day that I realized I needed to move to Portland and attend OHSU. I want to thank my friends and family, especially my wife, Angela, for all her love and support. And I want to thank the DMI students, faculty, and staff for all their help as well. And finally, I'd like to acknowledge my National Library of Medicine training grant, which funded my training for my PhD over the last 5 years. And with that, I'll leave it open for questions. Thank you.
Great, thanks, Julian. Very nice presentation. So we have some time for questions from the audience, either speak up or in the chat box.
Any questions? I'm not sure if I can see the chat box.
Yeah, no, there's no questions in the chat box. Well, maybe everyone fully understands.
I hope so.
Hey, Julian. There was something on slide 7 I wanted to ask you about.
Slide 7. Okay.
If you could pull it up.
Yeah. So just give me a second. Here, I'll do it. Sorry, yeah, just a second.
No worries.
Is this what you're talking about?
Yeah, I wanted you to explain that thing called the adjacency matrix a bit more to me.
Yeah, so this is how we infer the network structure. So, as we know, so the nodes of the network would be genes, or in my case, or splicing variants, and we don't know the underlying structure of the network. So essentially these edge weights, like how strong all the relationships of the genes are between the nodes of the network. So we need to infer that information. And so what we do is we use the expression data. So here's like an example of what the sample data might look like. And in actuality, we would have thousands and thousands of genes and maybe hundreds of samples. So we need to infer basically pairwise relationships between all the possible edge weights. So the adjacency matrix is it's a symmetric matrix of all the genes or the network. And so basically for every pair of genes, we try to infer a relationship. So kind of the most simple, kind of straightforward way that's commonly used is basically just to use like a correlation. So we can use like a Pearson correlation, and we see like as gene 1, how correlated is gene 1 with gene 2. And that then serves as sort of the initial value in the adjacency matrix. So that's just there's actually a lot of ways to kind of compute similarities. I actually used a different type of correlation measure called bi-weight mid correlations. And then what we could do is you'll also notice that there's actually negative values in here. So it doesn't necessarily make sense to have negative values in the network, at least for this approach. So usually we do some sort of transformation. So we can actually, in my case, I created what's known as a sign network, where I actually scaled the values where the negative correlation. So if it's a correlation of -1, we scale that to 0. The correlations of 1 stay at 1, and correlations of 0 go to 0.5. So it just shifts. So basically, it promotes the positive correlations, and then the negative correlations would be considered weak correlations. But this essentially it represents the weights of the edges in the network. We could also use a different approach where we may put a threshold on the correlations, and there's different ways to do this, where we could just create a binary network, and we only include edges if we have strong enough correlations. Does that answer your question?
Yeah, very good. Thank you.
Yeah. Okay, anybody else? Any other questions?
Great. Well, if not, we can excuse the public, myself included, and leave you behind with your committee who likely has further